then you can get an appreciation of just functionality of cells. And I give this lecture every year, but obviously this year it probably is even a little bit more relevant considering that we have this COVID crisis that we're experiencing and that vaccines are now being discussed really strongly. And as an immunologist, I'm still a little bit cautious on really what the outcome is going to be from these vaccines. We can talk about that, if not today, definitely in the spring when we get into immunology, because I teach in that course as well. Can everybody hear me pretty well? Thumbs up? Good. Okay. So what I want to do is I want to kind of blend in what we've talked about, what Dr. Hogan's talked about, what I've talked about in the previous lectures, so you can see how cell biology figures prominently into vaccines and the whole premise of it. And as future clinicians, of the many different tools you have at your disposal as practitioners, vaccines are one of the most important tools. Now, I'm not saying they're all perfect, they're not, and they aren't. And there's many facets of how vaccines work, and that's obviously something we'll talk about in the spring. But I wanted you to kind of look at it from a cell biology perspective on how vaccines are utilized, obviously the goal, but more interestingly is how these specialized cells that fall under the window of the immune system work together to respond to these vaccines, and with the goal of potentially helping the individual in disease processes. So that's kind of the focus of today's talk. And if there's things that I'm saying that don't...
Hello, I was on one Zoom.
All righty, so I'll move forward. So please, if you don't mind, just mute yourself. So why do we vaccinate animals and people? I mean, and I think there's some clear ideas out there, you know, and everyone's going to say basically protect the individual, the animal, from disease. That's true, but it's not the only reason. One of the other reasons, and obviously equally important, is that we recognize early on that everybody, animals and people included, have various responses. Their immune systems have various degrees of responses to the environment in the form of pathogens. And vaccines are no different because they're just specialized proteins that are packaged in some sort of slurry or matrix, and they're injected into an individual. And the idea is to promote an immune response against the different components of that vaccine. But as I said before, individuals, now all of you have been vaccinated for rabies, and I'm pretty sure at least most of you got 2, maybe even 3 vaccines, right? And have you all had titers done yet? Not yet. Okay, that will be coming, I guess, in the near future. But the thing that if you haven't appreciated, I want you to begin to appreciate is that not all individuals, although the vaccine constructs are the same and they're provided other variables, that is the administration of the vaccine is appropriate, storage of the vaccine is appropriate, individuals can respond differently to the vaccines, and the level of immunity is different as well. So vaccination not only is designed to protect the individual that receives the vaccine, but it also is designed, if you will, to create a biologic barrier and protect those individuals that don't have an optimal response to the vaccine. And this term we'll talk about is called herd immunity. I'm sure you're starting to hear that. You haven't heard it before in the literature, also and in the media, although that is also a point of contention at this point. So vaccination, that's the premise of what we do, is we're trying to prevent diseases from occur. Now, a number of you, and I've been reading the reports, and like I said, I'll be done. My goal is to get done by Wednesday, but probably Thursday. I'll be releasing all the grades for those as well. And the quiz is going out today as well. But a number of you wrote about certain tumors and talked about vaccines against tumors. And we won't go into too much depth about that, but the important thing about tumor vaccines, which is a little different from vaccines against pathogens like viruses and bacteria, is that in order for tumor vaccines to work, they have to have conserved proteins that are expressed on those tumors, regardless of the individual that has that particular cancer. And as many of you realize, if you write in your reports, that isn't always ideal or optimal because each individual has a unique tumor, and etiology factors into that. But there are certain viral tumors that have conserved epitopes in which vaccines are successful. So vaccines can be used against specific cancers. And one in particular that obviously is relevant to humans and to you all as well, because you're in that age group, is the human papillomavirus and the vaccine against that, which is Gardasil, Gardasil, for that particular virus. And get a chance, read up on it. It's kind of interesting. So that kind of sets the stage, if you will, for why we vaccinate. So how do we administer is the next question. So some of the things that, and don't worry about the angles, because I'll be honest with you, it's a neat picture. I don't think any of us in the veterinary world really worry about the angle which we do an injection, but there is some very important information on this particular slide to lay some groundwork that you need to remember. Notice I said remember, not memorize. And that is there are a variety of different injection sites we inject into an individual, an animal, or for people. IM sub-Q are the most common routes that we do with vaccines, and we'll explain that. You don't even have to memorize, it'll be a logic to it. Intradermal, typically we use for doing skin testing. That's a challenge. I'll be honest, I don't know how many of you have done it. Maybe some of you have been techs or licensed techs have done it. I've done it in dogs, and also have done this in mice because we've used mice for a lot of our other testing. It's a challenge doing an intradermal skin test. IV administration is never performed with vaccines. And the question is why? Well, IV administration gets right into systemic circulation. So if you want anything cleared quickly, or acted on quickly, you give it IV. Antibiotics, come to mind, certain drug therapies you give come to mind. The only other challenge with that is with IV administration, you gotta be careful because if there is systemic involvement, if there is any potential for an anaphylactic response, that's gonna be potentiated significantly by getting it into the intravascular system. But I think most importantly with vaccines, and this title says common routes of vaccine administration. You could also say common routes of drug administration to some degree as well. But going back to IV, it's cleared too fast. And that's an important thing when we think about a vaccine. One of the hallmarks of good vaccines is when you give it, it's going to be around for a while. So it's very important where the source or site in which you deposit that vaccine is crucial to make sure it's released very slowly. And we'll talk about that as well. But typically subcutaneous and IM injections are the common route of vaccine administration. So when we think about how these vaccines get in, and we're just look at the cells. Your immunology will factor in much heavier, like I say, in the spring. So we have two major arms of the immune system. So we have two major groups of cells that we focus on. They have similar functions. They proliferate. They release cytokines. They can mobilize through the body, some more so than others. But what sets the innate immune system apart from the acquired or adaptive immune system is these cells don't need priming. So that is, they're ready to go once they mature, okay? And they get into the sites wherever they find in the body. So ultimately, we know that the big purpose of the immune system is to protect the host from infection, protect the host from disease. So this obviously is foreign antigens being the major factor, and tumor cells can be foreign. So when neoplastic cells become neoplastic, they provide novel protein antigens or tumor antigens on their surface that make them targets for the immune system. The innate immune system is the first layer of defense, first bond. And we already talked a little bit about macrophages and dendritic cells, but similar to the acquired immune cells, which we'll talk about in a couple minutes, the B and T cells, these are bone marrow derived, so they originate from the hematopoietic compartment, and they migrate to the tissues where they reside and perform their function. The macrophages and the dendritic cells, and I'm hoping as I'm talking about this now that you've had histology, these set some little light bulbs go off. These are the phagocytic cells. These are the ones that have the capability of engulfing foreign antigen, vaccines being part of that, and then presenting it and processing it and putting on their surfaces and presenting it to the adaptive cells. Macrophages are the more aggressive, more efficient phagocytic cells, but dendritic cells are the more efficient antigen presenting capability, and that's because of their structure. As you can see here on my cool little slide that I included here, this is a dendritic cell. As you can see, there are a lot of appendages of the cell that allows it to present lots of antigen in the form of peptides, proteins, processed to many cells, where the macrophages here don't have this, even though they're similar in size, they don't have the same surface area. So that sets them apart. So dendritic cells are just more efficient antigen presenting cells. And that's the way nature's designed it. The neutrophils, basophils, NKs, mast cells, and eosinophils will say for the spring. The neutrophil is just to give you a perspective, and this is general information. I call them the kamikazes because these are the first cells to get there. They are phagocytic, but they die. They go in there and they try to create a lot of chaos at the site where infection is to make it difficult for pathogens to actually take hold. And then that whole process allows other inflammatory cells to come in, and then the inflammation ensues. The hallmark of innate immunity is they don't need priming, but there's an additional caveat. Although they don't need priming, they also don't generate memory. And part of the idea of vaccination is to create an army of cells, and these, you're going to learn to appreciate this later on. These are going to be B cells and T cells that are able to be ready when the animal or person is exposed to the pathogen, and be able to neutralize it before it has a potential for that pathogen to take hold in the body. So what I thought I would do is we're going to walk through, from a standpoint of cell biology, once again, walk through a vaccine being administered and how these cells interact with that particular vaccine and how that immunity is established. So looking at us, and this is the first slide, and we already talked about any potential compromise of the epithelium or the dermis layers, it can damage the cells, is going to lead to some degree of necrosis. So just sticking a needle into the site is going to cause some localized cell damage, and you're going to get some localized necrosis that occurs, but it's small, very minimal compared. And that's something I probably didn't explain as well as I should have, and that is necrosis. Cell death occurs, obviously, apoptosis necrosis, and we know why apoptotic cell death is preferred over necrotic cell death, because there's no inflammation. You can't help it when you stick a needle into an animal or into a person. You're going to get that localized, but it's so small that the cells there are able to repair, control the damage, control the inflammation, and minimize it. But if within that particular administration an infection ensues, well, it's a totally different story. That's why it's important we clean the site before we inject into the tissues, because we want to minimize any potential infection to ensue where our needle goes in. But let's say, for the sake of argument, everything goes well. Our needle goes in, and we're in the subcutaneous. Obviously, this is the smooth muscle. And we inject our vaccine, which is what you're going to learn in the spring, the different types, and we won't go into that today. That vaccine takes hold and establishes itself. And the way I designed this is my little cartoon. This shows that it actually seats in the smooth muscle, okay, and sits there. We call that the depot effect. And the reason we call it the depot effect is when the vaccine takes hold, it establishes itself. It's isolated a lot from a lot of the inflammatory cells. Dendritic cells are there as well, but it's not as well, what's the word I'm looking for, as accessible to the inflammatory cells right away. But even that, with even the complex, the matrix of the vaccine, it can create a sort of a deposit, if you will, of different proteins and components of those proteins that allows it to sit. And then it releases these proteins in a very controlled manner, okay? So it stores it, which is kind of like a depot, and then allows for sustained release for a long period of time. And this is really one of the key advantages of providing that vaccine at a good location. You want it to be able to sit and not be accessible as you would do like an IV injection to inflammatory cells. So what happens is, as these proteins, as the components of the vaccine are released into the tissues, the smooth muscle and the subcutaneous tissues, then these inflammatory cells, like the dendritic cells, can pick up these particular protein conjugates and peptides, whatever is being composed, engulf them, and then process them and put them on their surface. And this is some of the hallmarks of dendritic cells. And this is just general information. Don't worry. Like I said, this talk, I want you to understand general concepts. It's not going to be something you're required to know for the final exam, except I probably will ask an extra credit question on it, just to drive that home, that message. So you got receptor-mediated phagocytosis. It's an antigen-presenting cell. Remember we talked about MHC proteins? I kind of alluded to that being one of the important proteins on not just macrophages or dendritic cells, but MHC is also found on all nucleated cells. So it's a way that nucleated cells can process and present peptide on its surface, specific peptides that are infective, like viruses that get into the cells, to alert the immune cells that, hey, I'm an infected cell, I need to die. Okay. But what's going to govern this whole process is really the construct of the vaccine and how stable it is. So how long can it exist in the tissues? How long it can have a sustained release? Now, if it's very poorly constructed and it's not very stable and it degrades real quickly, then the level of immune response is going to be diminished because the dendritic cells and maybe inflammatory cells can clear it quickly. So what's going to govern that, just to drive home the message of the depot effect, is you want a vaccine that'll last in the tissues for a sustained period of time. So there's a controlled release, so you end up getting more inflammatory cells to the site and amplifying the inflammation, if you will. And typically, if you remember when you're vaccinated, if it's a good vaccine, you actually get a little granuloma type lesion there at the site of the injection. So for the sake of argument, the vaccine was administered properly, all was said and good in the world, it's a good vaccine. The dendritic cells are activated as well as macrophages, and they wall it off, and you get a little granuloma that forms. And it stays sustained. At this point, whatever signals occur, the dendritic cells can then migrate to the draining lymph nodes, which you've already learned in histology are situated, and anatomy, throughout the whole body. So this network of, basically, I call them immune forts. That's the way I look at them as an immunologist. They're all connected through this pathway known as lymphatics. And they come into the lymph node, and there's my cool lymph node, and obviously you don't even know this. Hopefully this is kind of a review. They come into the efferent ducts, and here's our little dendritic cell that I drew. And then you've got, this is the cortical region, just to remind you all. This is the medullary region, and these are known as follicles. And within the follicles are these germinal centers. And you don't need to know anything right now, but it's nice to have the terminology applied so you can see it from a cell standpoint. So as these dendritic cells migrate in, they'll hone in, if you will, on the lymph nodes. At the region of the follicles, they encounter these different cell clusters. Now, these follicles are rich in B cells. This is where our B cells are found. And surrounding these B cells in the paracortical region, which is what they're defining here, are the T cells, heavy presentation of T cells throughout. And then even there, within these follicles are what we call follicular dendritic cells. So there's special dendritic cells within the region of the lymph nodes that actually can trap free antigen that gets through the lymphatics and processes. So the immune system, nature has an interesting way of being able to allow trafficking of these cells at the site where in the periphery to the lymph nodes, or allow protein or peptides to get in, vaccines, whatever, to be trafficked through the lymphatics and be trapped and tagged by these follicular dendritic cells. And it's these cells that then present these processed peptides to the B and T cells to stimulate them. And we're going to look at that in just a minute. And then become activated. And sometimes, and because of lymphatics, they actually can migrate to other lymph nodes and activate other B and T cells as well. The important thing, and I know this is the big picture, I just want to kind of lay down the groundwork, is that in every lymph node, there are different clusters of B and T cells that have on their surface different receptors that recognize specific types, different unique types of peptides. That's nature's way of ensuring the ability of the immune system of the host, the animal, to recognize countless numbers. I don't know, billions, millions. I don't think the number is important, but recognize countless numbers of different epitopes. And in order to ensure that those B and T cells can be stimulated, the lymphatics are connected to ensure that those cells can migrate. Those dendritic cells, those T cells, those B cells, and even the proteins can migrate to the lymphatics to ensure that an immune response will occur. The caveat with vaccines is that still you have to have a sustained response to stimulate this. If the vaccine is neutralized too quickly or is degraded, then you won't get a good adaptive immune response. And we're going to look at that in more particular. Any questions? I can't see the chat. So, okay, I'm going to move forward. And Gina, I know I saw you there too. So keep an eye on the chat if you can see the chat window, because I can't. And if there's a question, just holler out. So let's look at the acquired immune system. Let's look at these clusters of cells. So we've got the innate cells that are from the hematopoietic compartment. Now we have this other collection of cells, which we know what they are. I mean, you've heard of them, but let's look at them just from a cell biology perspective. I just want to come at it at a different angle. So the acquired immune system consists of specialized cells called lymphocytes, okay, lymphocytes. We have B cells and T cells. B cells for either bone marrow derived, if you're mammals, bursal derived if you're a bird, and then a T cells is thymic derived, and that's for both, both mammals and birds. Although a lot of the, just a little bit of tidbit of information, a lot of the initial work that was done on understanding the immune system was done in chickens. And that's where B cells came out from bursal, believe it or not. I don't know if you guys did much anatomy. I guess you haven't done any anatomy on birds, but we can talk about that for another day. Now the B cells are following the window of humoral immunity, and they're produced primarily, they produce cytokines too, but their primary product are antibodies. B cell or humoral immunity is very effective at neutralizing foreign antigen outside the cell. Okay, so these cells are specifically targeting things that are outside the cells in the microenvironment. So extracellular, we call them extracellular infections. But they're also good and effective at preventing attachment of viruses to the cells. If those antibodies can recognize and bind to the ligands or proteins that bind to a cell before the virus gets in. Hence, one of the targets of proteins that we do in vaccines is to try to find specific epitopes or protein peptides that are expressed by viruses that allow them to link up to cells and infect those cells. T cells, also known as cell immunity, we already talked briefly about that. I kind of introduced it. And I know you've had it probably in micro as well. The helper T cell function is what we call the CD4s. There's different types of T helpers. You'll learn about that in the spring. And the cytotoxic, I know I talked about that as well. Those are the killer cells. Those are the extracellular killer cells, and the ones that can bind to an infected cell, release their perforins and granzymes, and move on. So these cells obviously are more effective in targeting abnormal cells or infected cells. So it can be a cancer. So and the thing is although we say T cells do a really good job, or their main job is targeting intracellular infections, that is infections or abnormal T cells, it's really in conjunction with the innate and the B cells. So all the immune system gets involved. You just have one particular arm that's more prevalent, more effective, if you will. But really is there's a total activation immune system. So both the humoral and sub-immune cells need to be primed before they can respond. And it's the acquired immune system, B cells and T cells, that are responsible for immunologic memory. And that's what the vaccine's targeting. That's what at least that's what we're using the vaccines for, is we want to build that army. So in a naive exposure, we may have one or two, that's not true, one or two, but say it's a few clones that are capable of recognizing what's unique to that vaccine. And truly, it's actually more than a few clones, but there's only one clone. But with vaccination, those clones multiply through mitosis, and we get an army of clones. Armies and armies, basically thousands and thousands and, hundreds of thousands, millions, whatever cells with proper immunization that have the ability to, when they are exposed, that animal's exposed to the particular pathogen, can deal with that pathogen very aggressively, and we'll look at that shortly. Now, just to highlight, this is a cartoon as well. You can see there's cytokines here. We won't get into the high affinity. We'll save that for the spring. But you can see, just to kind of tie it in a little bit. So these are, what I want to emphasize is, and you've done this before, and we'll have some fun with this in the spring as well, is when you look at lymphocytes, when you look at leukocytes, and I don't know if many of you had an opportunity yet to look at blood smears and do blood smears, but that's something you'll be doing, and the goal is to make you very proficient at. But these cells, when you look at them, just using H&E stain or modified right or not H&E, right? Modified right H&E, and then the Dipquick stain, they just look like lymphocytes. They're just round circular cells with big old nucleuses. And you can't tell a T cell from a B cell, although in peripheral blood, T cell is the predominant lymphocyte in peripheral blood. So you really have to, that's where flow cytometry comes into hand, when, especially if you're interested in being a clinical pathologist. We can actually identify these, and we'll look at them. But here, just to give you an idea, what this is supposed to show you, this is a T cell receptor on the cytotoxic T cell. This is a T cell receptor on the helper cell. And then these are antibodies, which we call B cell receptors. So B cells are kind of unique in the sense that not only do they secrete these antibodies, but when they're either in memory or when they're in a naive state, they express these antibodies on their surface loosely as B cell receptors to be able to recognize the antigen. And then upon activation, which is really cool, they release them. These are the only cells that can release these antibodies from the surface, and they become actual functional antibodies. So let's look at this a little bit closer. So we're going back to the needle going into the tissue, and let's say, for the sake of argument, this is subcutaneous tissue. The point I want to make here, when you look at this, is the injection. The vaccine can have a lot of different proteins that can induce inflammation, but the actual damage to the cells themselves can release different proteins. We call them DAMPs, and PAMPs. You'll learn about that in the spring. But what it's interesting is we think about just the dendritic cells, but in truth, not only do we see the dendritic cells playing a major role in the initial stage of activation of the innate immune system, but you have a lot of other cells present, like fibroblasts. This is called a plasmacytic dendritic cell. Don't worry about it. You got your tissue resident macrophages, but you also have the cells themselves that promote an inflammatory response. The actual injection itself can initiate a response. And you get all these different secretions. So you have different cytokines that are released, and their whole goal is to create an inflammatory response to attract inflammatory cells to neutralize whatever it is that's causing it and clear it quickly. Okay. So that's the case of this particular. So those are the first lines of defense. So upon activation, and we're saying for the sake of these are our peptides and our conjugate, the dendritic cell then migrates to the lymph node again. So this is cortex. This is a simpler diagram, if you will. And you can see it has the ability, and since this is the dendritic cell, these are all the different appendages, to simultaneously, depending on what cells it encounters. And the caveat is that when that dendritic cell presents a processed peptide on the surface, there's got to be a cell, T cell or B cell, that's in this case, we're looking at T cells, capable of recognizing that peptide. It's not automatic. They're kind of like bumper balls, if you will. They're bumping into different T cells and trying to express on their surface. And it's not just this particular, just two. This makes it oversimplified. You've got a huge cell that has many appendages. So the goal is really to stimulate many cells simultaneously as quickly as possible. But what I wanted to include, because this is a really cool from Nature Review, is it just shows you all the different permutations. One of the things that you're going to need to remember when you get into spring is that cytotoxic T cells have TCRs, T cell receptors. So does the CD4s. But these T cell receptors can recognize the same peptide, but it has to be presented by a specific MHC. So CD4s only recognize that peptide when it's presented by the antigen presenting cell via MHC class 2. And this is called MHC restriction. You'll learn more about in the spring. Cytotoxic T cells can only recognize processed peptide when it's presented via MHC class 1. So it's a very unique way. And as you already remember, all nucleated cells express MHC class 1. So it allows this particular guy, if you will, one of two, to go about looking at different cells, sample the cells, and examine them to make sure that they're not presenting a foreign antigen. So it's kind of an interesting novel case. When this one gets excited, you can get in the presence of bystander cells. It undergoes activation. It can become a killer cell, but it also can undergo mitosis and create more memory cells. This gives you a little bit more clarification distinguishing what naive CD4 T cell can undergo. Same story. It gets activated. It gets stimulation from other bystanders. We call them accessory cells as well. Not a big deal right now, but look at the interplay between the different cell types and how these cells become activated. That's kind of the cell biology, if you will. You have juxtacrine signaling, cell-cell contact. You get the release of cytokines, and then these cells, as they become activated, undergo differentiation is the term we use to become different types of T helper cells. You know, T helper 2, T helper 17, Th1. You'll learn about that. And we characterize them based on the cytokines that they can produce. IL-2, interferon gamma, when they be activated, become effectors, IL-17, IL-22, all different cytokines. So this is how we differentiate. And inclusive of that, we also look at different cell signaling pathways, which we kind of talked about, the JAK-STAT pathways. Okay. Now, T helper cells are also pivotal in activating B cells. B cells can activate independent. That's kind of neat about them. You'll learn about that. But they also can guide the different type of isotypes that are produced. And we won't go into too much detail at this point because I don't want to cover all immunology in less than 40 minutes. But you can see from even that, when B cells become clonally, we call them, they become excited, they polarize, they can clonally expand to produce more memory cells. This cartoon doesn't do it justice. You'll get more memory cells, or they can terminally differentiate into that plasma cell I was alluding to in one of my earlier talks. And this cell doesn't divide anymore. This cell basically is just an antibody farm. It's big. You've probably seen it in cytology, maybe histo. They kind of look like big sunny side up eggs because they're producing lots of protein. Their cytoplasm is huge. And they got a lot of mitochondria, a lot of ER, because they're undergoing transcription, they're undergoing translation, post-translation modification, and they're churning out all these different types of antibodies. Now, this is kind of misleading because it's telling you of different isotypes. When this plasma cell differentiates to become a protein-producing or antibody-producing farm, it's only going to produce one isotype. And we'll talk, like once again, you'll learn a little bit more. But stepping back, looking at it from the big picture, look at all the different interactions between different cells to get this to occur. That's the big message I want you to appreciate as Proby student doctors. It's a very complex process. It's very impressive. And we really try to understand it. If you begin to understand how this occurs, when you get into the disease processes, I think it'll be I know it'll be much easier for you to understand how these disease processes occurs when these particular, what I call novel optimal functions aren't occurring properly or occurring too high at a higher level than they should. So we're going to look at the T helpers, we're going to look at the cytotoxics real quickly, and then we're going to look at the B cells. So this kind of shows you some of the important things that we talked about previously as well. So we have an antigen presenting cell, and honestly, this kind of looks like a macrophage. It doesn't look like a dendritic cell. It's presenting, once again, it's T helper, so it's got to be MAC class 2. Here's your CD4, kind of like we saw with the cytotoxic CD8, binding to it, stabilizes the interaction, activates it, it polarizes. It secretes cytokines, like IL-2, to cause it to undergo clonal expansion, mitosis. So it's an autocrine signal. But antigen-presenting cells can also stimulate when they have that interaction, can release through the regulatory pathway, bring that back up, IL-2 as well. So you have both the paracrine and autocrine signals. Activation occurs, you're going to get effector cells. Those are T helper cells that are mature, doing their thing, they're secreting cytokines, or you're also going to get clones that basically expand to become memory cells, and they're not active. That's the army that we're talking about, that clonal expansion. The caveat with T cells and B cells is when those clones expand, they, on their surface, if it's a B cell receptor or a T cell receptor, they're all identical, and they recognize the same protein epitope that caused them to be activated. And that's good. It's an important component. That's what nature does, which is really cool. Now, the cytotoxic T cells, same story. So here we're looking at an antigen presenting cell. They did a blow up showing you the MHC class 1 on the surface of that little sucker there. There's your peptide. There's your T cell receptor. There's your CD8. It's a different diagram. Activation occurs. You get clones. So you get memory expansion of the cytotoxic T cells. They're not active. Then you also get effector cells. And for the sake of argument, let's pretend this should be apoptosis. So these are like apoptotic bodies. Looks like they kind of ruptured it, but no, it's undergoing apoptosis. It kills the cells through cell-cell contact. And this is an important concept. I think part of the challenge as veterinarians, and also, I mean, this is such an important area of our medicine when you think about it, because not only is vaccinology important, infections important, understanding the immune system is critical. Neonatal immunity, we'll get into that later on in spring, but so many facets of your daily life, immunology is, and understanding the immune system is critical. Okay, so that kind of gives you an idea. Any questions about that? No? Okay. I'm looking at Jeff's head and I'm looking at Gina. She's smiling, so that's cool. We're good. All right. So here's the B cells are the same. And the nice thing about this slide, it kind of gives you an appreciation of the whole vaccine concept. The initial exposure of the vaccine to a host, two-legged or four-legged, is what we call the naive exposure. So we're saying, for the sake of argument, the animal has never seen the components, that have never seen the pathogen, so that what the vaccine's presenting are unique peptides that simulate, in theory, all the major components, or most of the immunodominant components of that particular pathogen. And that first exposure is what we call the primary exposure or primary response, similar to what you had for rabies, because none of you theoretically had been exposed to rabies. So when you're vaccinated, that first vaccine was your primary response. What's nice about this slide, it shows you that in the germinal centers, in the follicles, you have lots of B cells. And when now the cool thing about B cells, they can recognize processed and they can recognize free antigen in the form of peptides. So what we see here with this diagram is we see, based on the color, what they wanted to demonstrate is that these are three different B cells that have on their surface B cell receptors that recognize, although it's not easy to appreciate, different peptides, different antigens, if you will, for simplicity. So this antigen has a triangular appearance, and this is what this is shown to demonstrate. And what we see here is when this B cell encounters that antigen, it can't recognize it, so there's no activation here. So the antigen comes along, and then the B cell with the red B cell receptors does recognize it, and it becomes activated. The B cell with the purple has a different epitope, or a different B cell receptor that can't recognize it as well. So it just drives home the point is you just got to realize and globally appreciate it, that when the B cells encounter these epitopes, they have to be able to recognize it. Not everyone will, which makes a lot of sense. But upon that initial activation, and you're going to get into a lot more depth about the specifics, and we get activation, we get clonal expansion, and we get armies of B cells. Some of them differentiate terminally, become the plasma cells. Others will produce memory clones. And this shows you, this one's showing also plasma cells. But we'll reach a certain stage where we get activation. This gets cleared, and all these peptides get bound. And after a period of time, once you have enough antibodies, there's a negative feedback loop. The production of antibodies start to wane. So we get that peak, and you're going to see it in one of my diagrams in just a minute. And then in the secondary response, which is the booster vaccine, you have lots more clones. And what you end up seeing is the same process all over again, except you have more B cells present, more memory B cells. So in theory, you get a much higher expansion, clonal expansion of that army of B cells. And that product, therefore, the amount of antibodies that we see numerically or geometrically increases, if you will, relative to the number of plasma cells that are produced. So the whole idea is to expand the army, but also expand the different products produced by that army. So you get increase in gene transcription, you get protein synthesis, you get post-translation modifications, so the cycle completes and continually arises until that particular vaccine is depleted in the body. And that's really the guide and the rationale. And we space it apart. You're going to learn about this 21 to 28 days between vaccines typically to optimize. And that's from just appreciating the time it takes for immune response to occur. To give you some appreciation, now what we're seeing here in this cartoon is just one clone. And in theory, the vaccine's goal is to activate many clones. But for one clone, we get a whole army if you will, a vast array of clone cells that recognize that particular peptide. And all these antibodies have the same, we call it configurations, recognize that same peptide. So they're all identical. So there's a massive clonal expansion. But in reality, with a vaccine, you want this to occur at multiple clonal levels. So you don't want a one clone or one monoclonal expansion, you want polyclonal expansion. So when we look at the big picture of how vaccines work and why is T cell and B cell clonal expansion the primary goal of vaccination, it goes back to what I said earlier, and that is that immunologic memory is a product of the adaptive or acquired immune system of B cells and T cells. So by taking one cell, you can recognize and respond to a foreign antigen, and making replicate cells or clones, clonal expansion, would greatly enhance the host's ability to protect itself from disease. But as I already said, it's not one cell. We want many clones, okay? And we recognize that that's the key. So an immune cell, that particular recognition, if you will, that clonal expansion is known as immunologic memory. What I alluded to in the previous cartoon is we showed one cell that replicated to produce identical clones. And that's a term that we call monoclonal expansion. But in reality, we want polyclonal expansion. That is, we want multitudes of different clones that recognize different protein epitopes. So that's what's important is that a vaccine has in its construct multitude of different epitopes, hundreds. And I honestly don't. It's one of the most closely guarded secrets in vaccine industry is really how these particular vaccines are made and how many different epitopes. And that's one of the things I'm interested in as an immunologist, just kind of pick through this to understand some very fundamental questions. And we'll talk about that toward the end. I'll tell you about it. But polyclonal expansion is really the key of vaccines because you really want a diverse array of different B cells and T cells capable of recognizing what you hope in the vaccine are specific protein epitopes that are common to the pathogen that animal will encounter, be it two-legged or four-legged. Okay. So the rationale for vaccination is both to educate and boost the immune system of the host against pathogens. Specifically, certain products, certain peptides of those pathogens that are known to be the peptides that cause a disease. You can generate an antibody titer for a vaccine. You say, I've created this vaccine against rabies or distemper. And I made it, it's new. I've used a unique formulation and I stick it in the animal and look at the titers I get. Or you can produce an antibody titer, but the problem or the challenge is you got to prove that titer is protective. Okay, and you do it, and one of the ways that you'll learn in the spring is they do neutralizing antibody titers. We'll talk about it later. But preventing disease, so one of the things for vaccination, going back to it, I'm sorry I got sidetracked. So that's the first bullet point. But if you think about it, and this is very true, going back to vaccination itself, the purpose of vaccination is to prevent a disease. And you think about it clinically as a clinician, as a practitioner, it is by far much more economically feasible, much more, has a better prognosis for preventing a disease than treating a disease, specifically for clients. So preventing a disease is a lot cheaper than treating it. And that's one of the arguments that you can always present as a clinician to clients who are surfing the web looking for reasons for not vaccinating the animal. Which brings up a good point. One of the great advances, current advances, technological advances is the internet. One of the greatest headaches that you're going to have as a clinician and a practitioner is the internet, because unfortunately there are people out there that feel very free to transmit their information and what they think is good or bad about science and medicine, specifically veterinary medicine. Human medicine is not immune either. And a lot of that data, a lot of that information is not carefully evaluated or challenged. So it falls on your shoulders as veterinarians, as practitioners, to be able to keep current with the information as you are challenged by the clients about the rationale for vaccines, the protocols, the efficacy of these particular vaccines. Immunologic memory is a function of a variety of different variables that you're going to learn as clinicians if you haven't already figured out some of them. One is the vaccine itself. Is it a good vaccine? What's going to determine that? Well, I think part of it is the companies it comes from. I mean, we vet them out pretty well, no pun intended. How well do you stable? How well is it stored? Is it properly stored? How well is it administered? What is the health of the animal? All those factors will play into the efficacy of a vaccine. The thing is, you need to be cognizant of that and start thinking about it now as future clinicians. Okay. Last, not every animal or person is going to be able to reach that maximum epitope, maximum antibody titer for protection. So really herd immunity, as we talked about in this little cartoon here, plays an important role. And even though there's a lot of issues out there, it's, you ask the question, there's something to think about as we go through it. Is it better to get exposed to disease versus not get exposed to disease? Well, obviously, if you lose COVID as our current question model, we do have people, individuals or populations that are not susceptible or less susceptible to this particular virus. And we know the age group, the age group as well as I do. Whether or not they should be vaccinated or not be vaccinated, that we are getting a larger population. Will they be able to develop herd immunity? With this particular virus, I don't know. It's up in the air because people still get, we've at least come across information that individuals who've had the virus can get the virus again. So it does come into the question of what level of immunity, because that's a natural infection. Shouldn't they have had the best level of protection because they actually had the disease, but they're getting sick again? So I don't know. There's some questions there that we have to kind of ponder and really ask about this particular vaccine that's coming up. So that was a side issue. We'll come back to that in just a minute. But just to finish out, how do we assess host immunologic memory generated by the vaccine? We measure the product, and we do it in the least invasive way. And that least invasive way is collecting blood. And we can either collect blood from the peripheral vein, usually jugular, cephalic, or saphenous, which we usually do. And you guys will be tested in the near future. And we look at either plasma or serum. And we look at the antibody titers. And that tells us. Now, what I did here, if you look at this particular, let me just slide this over for a second. I pulled this from an article. I forgot where it was a while back, but this shows this study. They were looking at two different sites of injection, intramuscular versus intradermal. And this was the primary, and this was the booster. And when they looked at titers, you could see with the intradermal, actually after the first booster, our cutoff is 0.5 international units per ml for protection for rabies. That's what it is. That's what if you didn't know that, that's what it is. And you can see through the intradermal route, the titer dropped below the protective level after 12 months. But if you look at the IM route, it stayed above, it wasn't a protective level, it stayed above 0.5 at 12 months, showing that just route of exposure can impact the antibody titer as well. But after the secondary booster, dexamethasone was comparable, but not as optimal as the IM. This study reinforced the fact that IM injection was much more efficacious for producing a sustained and strong titer than the intradermal route. And last, okay, what did I do? That was interesting. Here's the last slide. So this gives you a different perspective, and I wanted to kind of give you an idea of looking at titers. This is looking at antibody titers. This obviously is not, it's like 0.5 is the cutoff. So you can see here, looking at dog 1 first. With dog 1, we see a couple things. One was the antibody titers varies from dog to dog. So when they come up, and then you can appreciate, here's our 28-day window. So we're going to see probably around 14 days on a primary exposure. I would say 7 to 14 days. We'll see a pretty good peak. And then once the vaccine's clear, the titer starts to go down. But you already have your memory cells present. And on the secondary response, if our immune system is working optimally, we'll see a very strong titer increase, and it stays sustained. So this is kind of the classical representation of a primary and secondary immune response in a healthy dog under optimal conditions to a specific vaccine. And this is IgG, and we'll learn more about isotype switching later on. But this is the systemic immunoglobulin that we are actually looking at to show systemic protection, which brings up another good point about the COVID, because that's a respiratory virus. But that's a discussion maybe in the spring. We'll see how it goes. But in this slide, we see dog number 2. It gets an IgM response, which is here. And then after the secondary injection, or the booster, it still has an IgM response, but we see no measurable IgG response. And if you remember, we talked about immune deficiencies when we're talking about post-translation modification defects. I talked about different immune deficiencies like IgM deficiency, IgA deficiency, or IgG deficiency. This animal here is only producing IgM, but not producing IgG. This animal is suffering from a T cell deficiency because it's not allowing the B cells to undergo isotype switching because T cells drive that response. So this is kind of a classic example of how we can differentiate different deficiencies based on titers. And then this is the rabies virus. I forgot really why I put this in here, except just to show you some of the proteins that are unique to the rabies virus that we actually develop in our vaccines against rabies, targeting these specific proteins. Let me just get out. Any questions? And I can see everybody right now. Let me see if I can shut this copy down.